Probably the most well-developed target right now is focusing on macrophages, and in particular a receptor that they express called colony-stimulating factor 1. For short, it's a CSF-1 receptor. And so there are multiple compounds, either in preclinical or clinical development, and they're showing they're good at depleting macrophages, at least in the trials. We're still waiting to see whether they show efficacy either alone or in combination. Well, I tried to break it down for us, since it was an education session, onto kind of three main concepts of studying myeloid cells, and it was really form, function, and context were the three main fields. So one was to understand what myeloid cell you're looking at, which can be quite hard because they're very plastic cells, and they take on new phenotypes depending on what tissue they're in and what context they're experiencing. And that was the second issue, was the context of those cells. Whether you're talking about them in colon cancer or lung cancer or breast cancer, their phenotype and their function will be unique. And then you have to deal with the fact that you're going to add in therapeutic modalities on top of that, and that will further change what they're doing. So they're responding to paclitaxel chemotherapy in general versus radiation therapy. You may see very different dynamics on the role of those populations. Well, we certainly expect that as a certain patient was undergoing different rounds of chemotherapy or radiation therapy or any kind of new checkpoint inhibitors, that the phenotype of those cells will change, and certainly their function will change. And we're really just now starting to dissect that out. But at least in preclinical models, they found that if you combine targeting of macrophages with the aforementioned CSF-1 receptor pathways, you can enhance response to checkpoint blockade, chemotherapy, radiation therapy. But the role of the cells in each of those circumstances will be unique. It was not the focus of our session in particular, but that is the way that everyone is looking. And so whenever we're studying any other molecule now, it's going to be in the context of, does this relate to checkpoint blockade, in particular PD-1 and PD-L1 therapy. And so there's been, I think, 2 papers now showing preclinical that targeting these pathways works with checkpoint blockade. There are a number of stories in development, some that we saw here at AACR, showing that you can see combination. There was a talk the other day showing we see combination in mesothelioma, for example, within the lung, and these kind of stories are coming to the forefront now. I mean, there are multiple trials ongoing with various drugs in various types of cancers right now. They're still, because of how novel it was, they're still waiting on results from that. At least safety profiles look promising. And so they are moving forward with those, but efficacy, none of that data is available yet. We see it usually in combination with other therapies. So with one exception, there's fairly ubiquitous been shown that we have to combine these targets with other therapeutic modalities to see efficacy. And that's, of course, in preclinical models. And so with all that in mind, we realize it's going to be one part of, of a package of therapies that are administered to patients. So, I mean, right now it's being tried with chemotherapy. It's being tried in prostate cancer with androgen deprivation. I'm not sure whether it's trying with radiation therapy or not. And it will be checkpoint blockade therapy in combination, either now or shortly from now, for sure. I think we're making substantial progress now in really trying to break down what the role of those cells are, and understanding that we have to use multiple combinations to target those with other therapies. And I think the field was held back by just technology to some degree, and as those have advanced, we've been able to dissect the populations out and understand their function at a much greater detail, and that's helped us to figure out what's going on. The future questions will be what's the best context to use them in, and things like the temporal administration. Should we be treating before therapy, in the middle of therapy, or after therapy long term? And which of those is going to be the best way to go forward? And I think that's the question where we're talking myeloid cells and macrophages, or any other therapeutic context. I think that theme has come up repeatedly in the context of checkpoint blockade as well.